Wnt signaling pathway. The Wnt signaling pathways are a group of signal transduction pathways which begin with proteins that pass signals into a cell through cell surface receptors. The name Wnt is a portmanteau created from the names Wingless and Int-1. Wnt signaling pathways use either nearby cell-cell communication paracrine or same-cell communication autocrine. They are highly evolutionarily conserved in animals, which means they are similar across animal species from fruit flies to humans. 3 Wnt signaling pathways have been characterized: the canonical Wnt pathway, the noncanonical planar cell polarity pathway, and the noncanonical Wnt/calcium pathway. All three pathways are activated by the binding of a Wnt-protein ligand to a Frizzled family receptor, which passes the biological signal to the Dishevelled protein inside the cell. The canonical Wnt pathway leads to regulation of gene transcription and is thought to be negatively regulated in part by the SPAT-SON gene. The non-canonical planar cell polarity pathway regulates the cytoskeleton that is responsible for the shape of the cell. The non-canonical Wnt/calcium pathway regulates calcium inside the cell. Wnt signaling was first identified for its role in carcinogenesis, then for its function in embryonic development. The embryonic processes it controls include body axis patterning, cell fate specification, cell proliferation, and cell migration. These processes are necessary for proper formation of important tissues, including bone, heart, and muscle. Its role in embryonic development was discovered when genetic mutations in Wnt pathway proteins produced abnormal fruit fly embryos. Later research found that the genes responsible for these abnormalities also influenced breast cancer development in mice. Wnt signaling also controls tissue regeneration in adult bone marrow, skin, and intestine. This pathway's clinical importance was demonstrated by mutations that lead to various diseases, including breast and prostate cancer, glioblastoma, type Roman II diabetes, and others. In recent years, researchers reported first successful use of Wnt pathway inhibitors in mouse models of disease. History and etymology. The discovery of Wnt signaling was influenced by research on oncogenic cancer-causing retroviruses. In 1982, Roel Nuss and Harold Varmus infected mice with mouse mammary tumor virus in order to mutate mouse genes to see which mutated genes could cause breast tumors. They identified a new mouse proto-oncogene that they named Intone integration 1. Intone is highly conserved across multiple species, including humans and Drosophila. Its presence in D. melanogaster led researchers to discover in 1987 that the intone gene in Drosophila was actually the already known and characterized Drosophila gene known as Wingless WG. Since previous research by Christian Nusslein-Volhard and Eric Weishaus, which won them the Nobel Prize in Physiology or Medicine in 1995, had already established the function of WG as a segment polarity gene involved in the formation of the body axis during embryonic development. Continued research led to the discovery of further intone-related genes, however, because those genes were not identified in the same manner as intone, the int gene nomenclature was inadequate. Thus, the int/wingless family became the Wnt family and intone became Wntone. The name Wnt is a portmanteau of int and WG and stands for wingless-related integration site. Proteins. Wnt comprises a diverse family of secreted lipid-modified signaling glycoproteins that are 350–400 amino acids in length. The lipid modification of all Wnt's is palmitoylation of a single totally conserved serine residue. Polarity is necessary because it is required for Wnt to bind to its carrier protein Wnt-WLS so it can be transported to the plasma membrane for secretion, and it allows the Wnt protein to bind its receptor. Frizzled Wnt proteins also undergo glycosylation, which attaches a carbohydrate in order to ensure proper secretion. In Wnt signaling, these proteins act as ligands to activate the different Wnt pathways via paracrine and autocrine routes. These proteins are highly conserved across species. They can be found in mice, humans, xenopus, zebrafish, Drosophila, and many others. Mechanism. Foundation. Wnt signaling begins when a Wnt protein binds to the N-terminal extracellular cysteine-rich domain of a frizzled Fc family receptor. These receptors span the plasma membrane 7 times and constitute a distinct family of G-protein-coupled receptors, GPCRs. However, to facilitate Wnt signaling, co-receptors may be required alongside the interaction between the Wnt protein and Fc receptor. Examples include lipoprotein receptor-related protein LRP5/6, receptor tyrosine kinase RTK, and ROR2. Upon activation of the receptor, a signal is sent to the phosphoprotein Dishevelled DSH, which is located in the cytoplasm. This signal is transmitted via a direct interaction between Fc and DSH. DSH proteins are present in all organisms, and they all share the following highly conserved protein domains: an amino-terminal DIX domain, central PDZ domain, and a carboxy-terminal DEP domain. These different domains are important because after DSH, the Wnt signal can branch off into multiple pathways, and each pathway interacts with a different combination of the 3 domains. Canonical and non-canonical pathways. The 3 best characterized Wnt signaling pathways are the canonical Wnt pathway, the non-canonical planar cell polarity pathway, and the non-canonical Wnt/calcium pathway. As their names suggest, these pathways belong to one of 2 categories, canonical or non-canonical. The difference between the categories is that a canonical pathway involves the protein B-catenin, while a non-canonical pathway operates independently of it. Canonical pathway. The canonical Wnt pathway or Wnt/B-catenin pathway is the Wnt pathway that causes an accumulation of B-catenin in the cytoplasm and its eventual translocation into the nucleus to act as a transcriptional coactivator of transcription factors that belong to the TCF/LEF family. Without Wnt, B-catenin would not accumulate in the cytoplasm since a destruction complex would normally degrade it. This destruction complex includes the following proteins: axin, adenomatosis polyposis chole APC, protein phosphatase 2A PP2A, glycogen synthase kinase 3 GSK3, and casein kinase 1 aceconia. It degrades B-catenin by targeting it for ubiquitination, which subsequently sends it to the proteasome to be digested. However, as soon as Wnt binds Fc and LR5/6, the destruction complex function becomes disrupted. This is due to Wnt causing the translocation of the negative Wnt regulator, axin, and the destruction complex to the plasma membrane. Phosphorylation by other proteins in the destruction complex subsequently binds axin to the cytoplasmic tail of LR5/6. Axin becomes dephosphorylated and its stability and levels decrease. DSH then becomes activated via phosphorylation, and its DIX and PDZ domains inhibit the GSK3 activity of the destruction complex. This allows B-catenin to accumulate and localize to the nucleus and subsequently induce a cellular response via gene transduction alongside the TCF/LEF T cell factor/lymphoid enhancing factor transcription factors. B-catenin recruits other transcriptional coactivators such as BCL9, pygopis, and parafibromin/Hyrax. The complexity of the transcriptional complex assembled by B-catenin is beginning to emerge thanks to new high-throughput proteomics studies. However, a unified theory of how B-catenin drives target gene expression is still missing, and tissue-specific players might assist B-catenin to find its target genes. The extensivity of the B-catenin interacting proteins complicates our understanding. B-catenin may be directly phosphorylated at SIR552 by ACT, which causes its dissociation from cell-cell contacts and accumulation in cytosol. Thereafter 14-3-3s interacts with B-catenin SIR552 and enhances its nuclear translocation. BCL9 and pygopis have been reported, in fact, to possess several B-catenin-independent functions, therefore likely Wnt signaling independent. Non-canonical pathways. A non-canonical planar cell polarity PCP pathway does not involve B-catenin. It does not use LRP5/6 as its co-receptor, and is thought to use NR-home, RIC, PTK7, or ROR2. The PCP pathway is activated via the binding of Wnt to Fc and its co-receptor. The receptor then recruits DSH, which uses its PDZ and DIX domains to form a complex with Dishevelled associated activator of morphogenesis 1-DAA-MON. DAA-MON then activates the small g protein Rho through a guanine exchange factor. Rho activates Rho-associated kinase ROCK, which is one of the major regulators of the cytoskeleton. DSH also forms a complex with racone and mediates profilin binding to actin. Racone activates J and K and can also lead to actin polymerization. Profilin binding to actin can result in restructuring of the cytoskeleton and gastrulation. The non-canonical Wnt/calcium pathway also does not involve B-catenin. Its role is to help regulate calcium release from the endoplasmic reticulum in order to control intracellular calcium levels. Like other Wnt pathways, upon ligand binding, the activated Fc receptor directly interacts with DSH and activates specific DSH protein domains. The domains involved in Wnt/calcium signaling are the PDZ and DP domains. However, unlike other Wnt pathways, the Fc receptor directly interfaces with a trimeric G protein. This co-stimulation of DSH and the G protein can lead to the activation of either PLC or cGMP-specific PDE. If PLC is activated, the plasma membrane component PIP2 is cleaved into DAG and IP3. When IP3 binds its receptor on the calcium is released. Increased concentrations of calcium and DAG can activate CDC42 through PKC. CDC42 is an important regulator of ventral patterning. Increased calcium also activates calcineurin and Ca-MKI. Ca-MKI induces activation of the transcription factor in fat, which regulates cell adhesion, migration, and tissue separation. Calcineurin activates TA-cone and NLK kinase, which can interfere with TCF/SS-catenin signaling in the canonical Wnt pathway. However, if PD is activated, calcium release from the is inhibited. PD mediates this through the inhibition of PKG, which subsequently causes the inhibition of calcium release. Integrated Wnt pathway. The binary distinction of canonical and non-canonical Wnt signaling pathways has come under scrutiny, and an integrated, convergent Wnt pathway has been proposed. Some evidence for this was found for one Wnt ligand, Wnt5A. Evidence for a convergent Wnt signaling pathway that shows integrated activation of Wnt/Catwo plus and Wnt/SS-catenin signaling for multiple Wnt ligands was described in mammalian cell lines. Other pathways. Wnt signaling also regulates a number of other signaling pathways that have not been as extensively elucidated. Such pathway includes the interaction between Wnt and GSK3. During cell growth, Wnt can inhibit GSK3 in order to activate mTOR in the absence of B-catenin. However, Wnt can also serve as a negative regulator of mTOR via activation of the tumor suppressor TSC2, which is upregulated via DSH and GSK3 interaction. During myogenesis, Wnt uses PA and CREB to activate MyoD and MIFIV genes. Wnt also acts in conjunction with RIC and SRC to allow for regulation of neuron repulsion during axonal guidance. Wnt regulates gastrulation when C-cone serves as an inhibitor of reponent pace in order to modulate the cytoskeleton during gastrulation. Further regulation of gastrulation is achieved when Wnt uses ROR2 along with the CDC42 and JNK pathway to regulate the expression of PAC. DSH can also interact with a PKC pathway, PAR6 and LGL in order to control cell polarity and microtubule cytoskeleton development. While these pathways overlap with components associated with PCP and Wnt/calcium signaling, they are considered distinct pathways because they produce different responses. Regulation. In order to ensure proper functioning, Wnt signaling is constantly regulated at several points along its signaling pathways. For example, Wnt proteins are palmitoylated. The protein porcupine mediates this process, which means that it helps regulate when the Wnt ligand is secreted by determining when it is fully formed. Secretion is further controlled with proteins such as GP-RON177 Wnt-less and evenness interrupted, and complexes such as the retromer complex. Upon secretion, the ligand can be prevented from reaching its receptor through the binding of proteins such as the stabilizers DALI and glypican-3 GPC3, which inhibit diffusion. In cancer cells, both the heparan sulfate chains and the core protein of GPC3 are involved in regulating Wnt binding and activation for cell proliferation. Wnt recognizes a heparan sulfate structure on GPC3, which contains IDO, ATWO, S and GLC and S6S, and the 3O sulfation in GLC, and S6S3S enhances the binding of Wnt to the heparan sulfate glypican. A cysteine-rich domain at the end lobe of GPC3 has been identified to form a Wnt binding hydrophobic groove including phenylalanine 41 that interacts with Wnt. Blocking the Wnt binding domain using a nanobody called HN3 can inhibit Wnt activation. At the Fc receptor, the binding of proteins other than Wnt can antagonize signaling. Specific antagonists include DKK, Wnt inhibitory factor 1 WIF1, secreted Frizzled-related proteins SFRP, Cerberus, FRZB, WISE, SOST, and naked cuticle. These constitute inhibitors of Wnt signaling. However, other molecules also act as activators. Norrin and R-spondin-2 activate Wnt signaling in the absence of Wnt ligand. Interactions between Wnt signaling pathways also regulate Wnt signaling. As previously mentioned, the Wnt/calcium pathway can inhibit TCF/B-catenin, preventing canonical Wnt pathway signaling. Prostaglandin Etwo is an essential activator of the canonical Wnt signaling pathway. Interaction of PG-Etwo with its receptors Etwo/Fr stabilizes B-catenin through cAMP/pKa-mediated phosphorylation. The synthesis of PG-Etwo is necessary for Wnt signaling-mediated processes such as tissue regeneration and control of stem cell population in zebrafish and mouse. Intriguingly, the unstructured regions of several oversized intrinsically disordered proteins play crucial roles in regulating Wnt signaling. Induced cell responses. Embryonic development. Wnt signaling plays a critical role in embryonic development. It operates in both vertebrates and invertebrates, including humans, frogs, zebrafish, C. elegans, Drosophila, and others. It was first found in the segment polarity of Drosophila, where it helps to establish anterior and posterior polarities. It is implicated in other developmental processes. As its function in Drosophila suggests, it plays a key role in body axis formation, particularly the formation of the anteroposterior and dorsoventral axes. It is involved in the induction of cell differentiation to prompt formation of important organs such as lungs and ovaries. Wnt further ensures the development of these tissues through proper regulation of cell proliferation and migration. Wnt signaling functions can be divided into axis patterning, cell fate specification, cell proliferation, and cell migration. Axis patterning. In early embryo development, the formation of the primary body axis is a crucial step in establishing the organism's overall body plan. The axes include the anteroposterior axis, dorsoventral axis, and right-left axis. Wnt signaling is implicated in the formation of the anteroposterior and dorsoventral DV axes. Wnt signaling activity in anterior-posterior development can be seen in mammals, fish, and frogs. In mammals, the primitive streak and other surrounding tissues produce the morphogenic compounds WNTS, BMPs, FGFs, nodal and retinoic acid, to establish the posterior region during late gastula. These proteins form concentration gradients. Areas of highest concentration establish the posterior region, while areas of lowest concentration indicate the anterior region. In fish and frogs, B-catenin produced by canonical Wnt signaling causes the formation of organizing centers, which, alongside BMP, elicit posterior formation. Wnt involvement in DV axis formation can be seen in the activity of the formation of the Spemann organizer, which establishes the dorsal region. Canonical Wnt signaling B-catenin production induces the formation of this organizer via the activation of the genes twin and SIAM-1. Similarly, in avian gastrulation, cells of the collar's sickle express different mesodermal marker genes that allow for the differential movement of cells during the formation of the primitive streak. Wnt signaling activated by FGF is responsible for this movement. Wnt signaling is also involved in the axis formation of specific body parts and organ systems later in development. In vertebrates, sonic hedgehog SHH and Wnt morphogenetic signaling gradients establish the dorsoventral axis of the central nervous system during neural tube axial patterning. High Wnt signaling establishes the dorsal region while high SHH signaling indicates the ventral region. Wnt is involved in the DV formation of the central nervous system through its involvement in axon guidance. Wnt proteins guide the axons of the spinal cord in an anterior-posterior direction. Wnt is also involved in the formation of the limb DV axis. Specifically, Wnt savina helps produce the dorsal patterning of the developing limb. In the embryonic differentiation waves model of development, Wnt plays a critical role as part of signaling complex in competent cells ready to differentiate. Wnt reacts to the activity of the cytoskeleton, stabilizing the initial change created by a passing wave of contraction or expansion, and simultaneously signals the nucleus through the use of its different signaling pathways as to which wave the individual cell has participated in. Wnt activity thereby amplifies mechanical signaling that occurs during development. Cell fate specification. Cell fate specification or cell differentiation is a process where undifferentiated cells can become a more specialized cell type. Wnt signaling induces differentiation of pluripotent stem cells into mesoderm and endoderm progenitor cells. These progenitor cells further differentiate into cell types such as endothelial, cardiac, and vascular smooth muscle lineages. Wnt signaling induces blood formation from stem cells. Specifically, Wnt3 leads to mesoderm-committed cells with hematopoietic potential. Wnt antagonizes neural differentiation and is a major factor in self-renewal of neural stem cells. This allows for regeneration of nervous system cells, which is further evidence of a role in promoting neural stem cell proliferation. Wnt signaling is involved in germ cell determination, gut tissue specification, hair follicle development, lung tissue development, trunk neural crest cell differentiation, nephron development, ovary development, and sex determination. Wnt signaling also antagonizes heart formation, and Wnt inhibition was shown to be a critical inducer of heart tissue during development, and small molecule Wnt inhibitors are routinely used to produce cardiomyocytes from pluripotent stem cells. Cell proliferation. In order to have the mass differentiation of cells needed to form the specified cell tissues of different organisms, proliferation and growth of embryonic stem cells must take place. This process is mediated through canonical Wnt signaling, which increases nuclear and cytoplasmic β-catenin. Increased β-catenin can initiate transcriptional activation of proteins such as cyclin DUN and c-Myc, which control the GON to S phase transition in the cell cycle. Entry into the S phase causes DNA replication and ultimately mitosis, which are responsible for cell proliferation. This proliferation increase is directly paired with cell differentiation because as the stem cells proliferate, they also differentiate. This allows for overall growth and development of specific tissue systems during embryonic development. This is apparent in systems such as the circulatory system, where Wnt3 leads to proliferation and expansion of hematopoietic stem cells needed for red blood cell formation. The biochemistry of cancer stem cells is subtly different than that of other tumor cells. These so-called Wnt-addicted cells hijack and depend on constant stimulation of the Wnt pathway to promote their uncontrolled growth, survival, and migration. In cancer, Wnt signaling can become independent of regular stimuli through mutations in downstream oncogenes and tumor suppressor genes that become permanently activated even though the normal receptor has not received a signal. β-catenin binds to transcription factors such as the protein TCF4, and in combination, the molecules activate the necessary genes. LF3 strongly inhibits this binding in vitro in cell lines and reduced tumor growth in mouse models. Prevented replication and reduced their ability to migrate, all without affecting healthy cells. No cancer stem cells remained after treatment. The discovery was the product of rational drug design, involving alpha screens and ELISA technologies. Cell migration. Cell migration during embryonic development allows for the establishment of body axes, tissue formation, limb induction, and several other processes. Wnt signaling helps mediate this process, particularly during convergent extension. Signaling from both the Wnt-PCP pathway and canonical Wnt pathway is required for proper convergent extension during gastrulation. Convergent extension is further regulated by the Wnt/calcium pathway, which blocks convergent extension when activated. Wnt signaling also induces cell migration in later stages of development through the control of the migration behavior of neuroblasts, neural crest cells, myocytes, and tracheal cells. Wnt signaling is involved in another key migration process known as the epithelial mesenchymal transition (EMT). This process allows epithelial cells to transform into mesenchymal cells so that they are no longer held in place at the laminin. It involves cadherin downregulation so that cells can detach from laminin and migrate. Wnt signaling is an inducer of EMT, particularly in mammary development. Insulin sensitivity. Insulin is a peptide hormone involved in glucose homeostasis within certain organisms. Specifically, it leads to upregulation of glucose transporters in the cell membrane in order to increase glucose uptake from the bloodstream. This process is partially mediated by activation of Wnt/β-catenin signaling, which can increase a cell's insulin sensitivity. In particular, Wnt10 is a Wnt-protein that increases this sensitivity in skeletal muscle cells. Clinical implications. Cancer. Since its initial discovery, Wnt signaling has had an association with cancer. When Wnt was discovered, it was first identified as a proto-oncogene in a mouse model for breast cancer. The fact that Wnt is a homologue of Wg shows that it is involved in embryonic development, which often calls for rapid cell division and migration. Dysregulation of these processes can lead to tumor development via excess cell proliferation. Canonical Wnt pathway activity is involved in the development of benign and malignant breast tumors. The role of Wnt pathway in tumor chemoresistance has been also well documented, as well as its role in the maintenance of a distinct subpopulation of cancer-initiating cells. Its presence is revealed by elevated levels of β-catenin in the nucleus and/or cytoplasm, which can be detected with immunohistochemical staining and Western blotting. Increased β-catenin expression is correlated with poor prognosis in breast cancer patients. This accumulation may be due to factors such as mutations in β-catenin, deficiencies in the β-catenin destruction complex, most frequently by mutations in structurally disordered regions of APC, overexpression of Wnt ligands, loss of inhibitors, and/or decreased activity of regulatory pathways such as the Wnt/calcium pathway. Breast tumors can metastasize due to Wnt involvement in EMT. Research looking at metastases of basal-like breast cancer to the lungs showed that repression of Wnt/β-catenin signaling can prevent EMT, which can inhibit metastases. Wnt signaling has been implicated in the development of other cancers. Changes in cTNN bone expression, which is the gene that encodes β-catenin, can be measured in breast, colorectal, melanoma, prostate, lung, and other cancers. Increased expression of Wnt ligand proteins such as Wnt, Wnt2, and Wnt7 were observed in the development of glioblastoma, esophageal cancer, and ovarian cancer, respectively. Other proteins that cause multiple cancer types in the absence of proper functioning include RO-RONE, RO-R2, SFR-FR, WNT5A, WI-FON, and those of the TCF/LEF family. The link between PG-ETWO and Wnt suggests that a chronic inflammation-related increase of PG-ETWO may lead to activation of the Wnt pathway in different tissues, resulting in carcinogenesis. Type Roman II diabetes. Diabetes mellitus type 2 is a common disease that causes reduced insulin secretion and increased insulin resistance in the periphery. It results in increased blood glucose levels, or hyperglycemia, which can be fatal if untreated. Since Wnt signaling is involved in insulin sensitivity, malfunctioning of its pathway could be involved. Overexpression of Wnt5, for instance, may increase susceptibility due to its role in adipogenesis, since obesity and type II diabetes have high comorbidity. Wnt signaling is a strong activator of mitochondrial biogenesis. This leads to increased production of reactive oxygen species ROS known to cause DNA and cellular damage. This ROS-induced damage is significant because it can cause acute hepatic insulin resistance or injury-induced insulin resistance. Mutations in Wnt signaling-associated transcription factors, such as TCF7L2, are linked to increased susceptibility.